I appreciate everybody coming tonight. We have Dr. Cade, who is here to explain a bit about his upcoming study for the characterization and metabolic phenotype in Barth syndrome with people who have had transplants. Dr. Cade has done this study in the past with healthy individuals and controls, and this takes this a step farther to include individuals who have had transplants. Todd, thanks for coming tonight, and you can go. Thank you.
Okay, well, thanks so much, Shelley, for just allowing me to go ahead and advertise and tell everyone a little bit about the study and the transplant population that we want to do. So what I want to do first, since I know a lot of you, but who are on the call, but I want to explain a little bit about what I do, what our lab does, and then how we even got started doing this line of research. And then I'm going to tell you about the big study that we pretty much are just finishing, that likely a lot of you have heard about already, the NIH-funded study that we did in the guys with Barth that did not have a transplant. So really, what this study that I'm going to talk about tonight is an extension of that NIH-funded study to really now look at the guys with Barth syndrome that had transplants. So hopefully everything will become clear. And again, if you have questions along the way, please just go ahead and I can see the questions that come up, but if you want it privately, again, send it to Shelley and she'll send it to my email, and we'll make sure that that gets answered. Okay, so what do we do here at Washington University? So first of all, we're not in Washington State. We're actually in St. Louis. It's Washington University in St. Louis. And so what exactly does our lab do, and what are we really interested in? But so overall, we're really interested in mechanisms or causes and treatments, including nutritional treatments, exercise, drug treatments for skeletal muscle and heart substrate abnormalities. When I say substrate metabolism abnormalities, I mean I'm talking about substrates or nutrients, including amino acids, glucose, fatty acids, things that that our bodies use for energy and cellular metabolism. So particularly, we are interested in Barth syndrome, as everyone knows. We also study things like HIV-related metabolic syndrome, diabetes, and obesity type research. And some of the research techniques are some of the things that we use to study metabolism in Barth syndrome. We're going to be talking about tonight, and then really most of these I'm going to be talking about in the context of the study that we're currently doing and that we're hoping to do in the transplant population, are things like exercise testing, including like endurance or aerobic exercise testing, and strength testing like resistance training. We also look at body composition and bone density. We're interested in how the whole body and skeletal muscle uses fat, sugar, and protein for energy. We can also look at how the heart specifically uses these nutrients for their metabolic needs. We also use MRI to look at actually how skeletal muscle and heart produces energy. So it's a really unique way to look at this. We also use tools such as cardiac imaging, which many of you, I'm sure all of you understand, echocardiography. We also use PET imaging, which we're not going to do in the transplant study. And then we do things like metabolomics. We look at a lot of the different metabolites in the blood and kind of see how they're different and how they're affected by Barth syndrome or training or some sort of drug treatment. So really, what are we interested in Barth syndrome? What aspects of Barth syndrome are we particularly interested in our lab? Well, we're really interested in why, what are the reasons for reduced heart function, skeletal muscle weakness and fatigue, why guys with Barth syndrome have severe exercise intolerance, and we're really interested in how energy production in the heart and skeletal muscle contribute to this, especially by the mitochondria, which I'm sure everyone knows mitochondria is the powerhouse of the cell, really the energy producing part of the cell that is affected in Barth syndrome. So we're really interested in learning more about this so we can develop treatments to hopefully help alleviate or correct these deficits in Barth syndrome. So how do we even get started in this line of research? This happened, we began these studies in 2006 at the Disney conference with the shuttle. I'm sure many of you were there with the shuttle launch, which I thought was really neat got a chance to see that. But really, why do we get this? Why do we start this research in the first place? Well, there's a lot of clinical complaints of high fatigue, physical activity intolerance, not able to exercise, not able to participate in sports and activities that their peers participated in. And we really didn't have an objective measure of fatigue and activity intolerance. We just had these reports and complaints. So we really weren't sure if this exercise intolerance, this fatigue, was caused by either the heart deficits or skeletal muscle deficits, or potentially a combination of both. So at this conference in Orlando, in conjunction with Barry Byrne and Carolyn Taylor, we basically did exercise testing and echocardiography in a number of boys, and also looked at their muscle oxygen use through this technique called near-infrared spectroscopy. So in that study, we studied 15 guys with Barth, and then their 9 controls, which really consisted of their friends and siblings. So this is a picture that was given permission to use. So this is kind of the exercise setup. This was actually not at the conference, but here at WashU. But as you can see, it's a pretty complex setup where we measure oxygen consumption through the mouth. You can see here this participant is breathing in and out. We can measure how much oxygen he's consuming and how much carbon dioxide he's breathing out, and measuring his heart function. And during exercise, we can tell a lot about his muscle and heart's ability to utilize oxygen, their aerobic energy, really the main or the gold standard of exercise testing or peak fitness. So what we found here, can everyone see my cursor if I move it around? Okay, I'm going to assume that you can. So on this axis here, this is volume of oxygen consumed. So this is basically like your aerobic energy that you're able to use, and this is work rate. So every number here, as it gets higher and higher, basically means it's harder and harder to pedal the bike as they're exercising. And as you can see, the open circles are participants with Barth syndrome, and the closed circles are participants who are unaffected, their peers and their friends. And as you can see, most guys with Barth petered out or pooped out right around between 60 and 80 watts compared to their peers, which were able to go up to 200 plus watts. So they're not able to generate as much force with their legs. And then their peak oxygen consumption or aerobic energy was significantly lower, right around 15 compared to about 35 of their peers. So really what this is saying is we were able to demonstrate that their peak fitness or peak exercise tolerance was significantly impaired when compared to their unaffected peers. And then what we also did, so we were again interested in what caused this exercise intolerance. Was it heart problems? Was it a dysfunction of the heart or the skeletal muscle, or a combination of both? So first we looked at heart function here, and this is basically ejection fraction, which I think is everyone is familiar with. And as you see, the controls here, when you pre and post was at rest and then at peak exercise. So if you normally when you have your heart function or your heart contractility goes up with exercise, and you can see that happens with the unaffected peers, the control group. But if you look at both groups here are with Barth syndrome, those are on cardiac beta blockers versus not. The reason why we looked at these in two separate groups is because we know that just the medication beta blocker can actually depress heart function during exercise a little bit. So that's what we looked at that. But as you can see, with exercise from rest to at peak exercise, heart contractility really didn't improve at all in either group with Barth syndrome. So really what that indicated to us was that the heart didn't have a good energetic reserve, was not able to keep up with the requirements of exercise. It was not able to increase its contractility with exercise.
Todd, for some reason we can't see your cursor when you move it around.
Okay.
So,
How about,
yeah,
how about on this
now
side?
I see it. Now I see it. Now we see it.
Sorry
Yeah.
about that. I'm on the wrong screen here.
Okay, we can see it now.
Okay. So then next what we did was we looked at skeletal muscle oxygen use. Again, these are just markers of how well the skeletal muscle can use oxygen. So just let me kind of walk you through this. Again, this is during exercise. The higher work rates means higher exercise intensity. And as you can see with the control group here, the closed circle, the oxygenated hemoglobin with increasing oxygen should actually decrease. And that's what you see what happens with the control group. But it paradoxically increases with Barth syndrome. What that really just shows us is that the skeletal muscle is not able to extract and use oxygen like their unaffected peers. So ultimately what that showed us is that both heart and skeletal muscle problems contributed to the exercise intolerance with Barth syndrome. I'm just going to actually skip over this. I don't think this is going to probably be more confusing than anything. But so this initial study in 2006 led to a lot of questions that we didn't really know in Barth syndrome. So we found out that severe exercise intolerance, we were able to quantify that in Barth syndrome. We were able to discover that this exercise intolerance was caused by a combination of heart and skeletal muscle impairments. And this was consistent with what we know about Barth syndrome. So we know that Barth syndrome affects the mitochondria and heart and skeletal muscle. So it makes sense that it affected both heart and skeletal muscle. It had very low exercise capacity. And what we also found that it looked like they had some substrate utilization abnormalities, which means that it looked like from the exercise testing that they were potentially not able to use certain nutrients appropriately. And then we also know that many of these boys have very low skeletal muscle mass. And we thought potentially that could be indicative of maybe protein metabolism impairments. So we thought, okay, we don't know a lot about substrate metabolism or nutrient metabolism, so maybe this would be something good to study in Barth syndrome. So at the time, we're using this complex methodology, which I'll discuss a little bit more in later slides. I was using this currently in other populations, and it would be an ideal methodology to really look at some of these questions in Barth syndrome and really see in Barth syndrome what abnormalities in nutrient metabolism could potentially be causing some of these heart and skeletal muscle impairments. And that really led to a Barth Syndrome Foundation pilot study that we were able to collect pilot data, which I'm not going to talk about just for time's sake. But that was able to provide us with some pilot data to apply for an NIH-funded study, which we did about 5 years ago. And this is the title of it, called Heart and Skeletal Muscle Metabolism, Energetics and Function in Barth Syndrome. And this was the study that I've talked to the conference the last couple of years about, and that we were able to recruit 60 total subjects, 30 with Barth syndrome, 30 without Barth syndrome, and really overall to look at some of these nutrient metabolism causes for heart and skeletal muscle impairments in Barth syndrome. So what we initially thought that the ability of the heart and skeletal muscle to use fat would be severely impaired in Barth syndrome, and basically, people with Barth syndrome would have to use increased protein for energy. Now, if you use increased protein for energy over time, a long time use of it, you potentially could break down heart and skeletal muscle because that's the stores of protein. And we thought that could potentially contribute to or be a cause of some of the skeletal muscle weakness, heart dysfunction that we see in Barth syndrome. So that's what we initially thought and initially hypothesized in this large study that we're just finishing up. And this is going to be very relevant to the transplant study. So basically, I'm going to describe a little bit about what our hypothesis, what our aims were in this large study, some of the techniques that we use to study this, because it's going to be very pertinent into the transplant study, because basically the transplant study is just replicating this exact same study that we just completed. So our first aim in this large study that we just completed was to characterize heart and muscle nutrient metabolism in a group of children and adults with Barth syndrome and compare that to unaffected peers who were matched for age, puberty level, and activity level. So we're able to study about 60 patients total, 30 with Barth syndrome, and of that 30 with Barth syndrome, 15 were children and 15 were adults. So what we did is we looked at skeletal muscle and heart, fat, glucose, and amino acid metabolism during this clinical research paradigm where they rested for 2 hours, they exercised for a half an hour at a low level, and then we measured them for an hour post-exercise. So we were able to look at how they metabolize fat, sugar, or glucose, and amino acids, or amino acids basically just make up protein or protein metabolism. So we use really these complex methodologies, which I'll briefly explain in a couple slides to look at this. And then our second aim was to really look at the relationship between their heart and muscle nutrient metabolism, which I just explained. And then how does this relate to the energy production of the heart and the function of the heart and skeletal muscle, again, in this group. And how we did that is we actually used MRI to look at energy production. We used echocardiography to look at heart function. And then we used exercise testing, which I just explained, to look at muscle function. And then as an exploratory aim, which I'm not going to talk much about, and through collaboration with Barry Byrne and Christy Pasek at the University of Florida, we're able to take blood cells and make inducible pluripotent stem cells, basically convert these blood cells into heart cells, and then study them basically in cell culture and in real complex molecular biology ways, which I'm not going to talk about. So a really neat way to, without getting muscle biopsies, to actually just get blood cells and make them into cardiomyocytes. And the next summer, Christy Pasek will be able to explain a lot about more of what we found in this study. So basically, this is a schematic of the clinical metabolism visit, which the transplant study will have. So what we did is, you can see here, we gave a little bit of glucose tracer and leucine tracer and palmitate tracer in these studies, and basically looked at these metabolism of all these nutrients over the course of 3.5 hours. Of these 3.5 hours, 2 hours was resting, and you can see we collected blood and breath samples. A half an hour of it was a low-level exercise, so 50% of their peak exercise capacity, and then 1 hour of post-exercise capacity. So I'll explain a little bit more, but basically a 3.5 hour study where we're able to collect blood and breath samples. So, and I'll explain a little bit more about how we did that. This isn't multiple sticks. We had a catheter in, so we're just able to draw the blood from an indwelling catheter. This is a picture. As you can see, one of our participants is extremely happy during this. So this is a very enjoyable study. So you can see here in this setup, this is a recumbent exercise bike. And as you can see here, these are the tracers that are being infused. And then, unfortunately, you cannot see his arm, but we draw blood from his his hand here. Can't really see that, but this is a warming box. All this does is warm the hand, so blood draws are a lot easier. Okay, and as you can also see here, is a blood pressure cuff. We take intermittent blood pressures, and then behind here is a metabolic cart, which we take. It's the same thing as when we do exercise testing. We basically take breath measurements of his oxygen consumption during exercise. So what in the world are stable isotope tracers? Okay, they sound crazy, they sound a little dangerous, but they're not. So basically, for an example of hydrogen, so our bodies mostly consist of hydrogen, carbon, and oxygen, and also a little bit of nitrogen as well. So our basically, organically, our bodies consist of this. The food we eat consists of this, and most of the hydrogens that we eat or that we're comprised of, and the carbons, our hydrogen basically has an atomic weight of 1. There are also naturally occurring hydrogens that actually weigh 2, and that's because they have 1 extra neutron, in addition to the protons and electrons in these elements. Okay. So when I want to be clear that these are naturally occurring, they're stable, and they're safe. There are also some radioactive hydrogens, so that would be 2 extra neutrons that have a little radioactivity. But when we talk about stable isotope tracers, we're talking about naturally occurring. I mean, occurs naturally in nature, in the foods we eat, in the composition that we are. They comprise about less than 1% of all the hydrogens in nature, but they are naturally occurring. Same thing with carbon. Most of the carbons that naturally occur weigh 12, but there's also stable isotopes of carbon that weigh 1 more, so has 1 extra neutron that occur in nature that weigh 13. And this is important because when we give these tracers that weigh 13 and 12, we can discriminate the tracer from the naturally occurring when we take blood and breath samples. So we're able to tell a lot about metabolism just because we can determine through mass spectrometry what is tracer and what is not tracer, because they weigh 1 more. again, totally safe to give. It occurs naturally in nature. And just to show you here, this is the structure of glucose and the structure of a fat. You can see lots of carbons, lots of hydrogens in both the fats and the glucose. And what it is, these tracers is there are a couple of the hydrogens weigh one more. Again, they weigh 2 versus instead of 1. Again, it doesn't really alter the composition of the glucose. It just means we can measure the tracer, the glucose tracer, because it weighs 2 more than the naturally occurring. And same thing when we label on the omega, the fatty acid on the carbon or the hydrogen. And then, so what we do is we take these tracers, we get them from a company that makes them. They get compounded. This is actually not exactly how it's done. I just threw this picture in, but basically they're compounded in a research pharmacy and sterility tested. So they're completely safe and sterile. And then, as you see here, this is a study. We infuse them into the participant, and by taking blood and breast samples, we can tell a lot about metabolism. Things like how sensitive are the tissues to glucose? How well are the tissues able to use glucose? How well can the patient utilize fat for energy? How much protein they're able to break down or to use for synthesis. So there's a lot of things we can tell by just using these tracers and taking blood samples and breast samples that we can learn about metabolism. This crazy device is just what we collect breast samples in. So the participant breathes in, air comes in, they breathe out, air gets collected in here. We're able to collect in these containers and then measure the tracers that come out in the breath. So basically, if the tracer is labeled on the carbon, when you breathe out, we can measure that carbon as it gets oxidized or burned as carbon dioxide. We can put it on a mass spectrometer and really accurately measure that. And then this is the device that we can measure that on. It's called a mass spectrometer. So I just wanted to briefly explain. I know this is very highly complex and probably a little bit difficult to understand, but I just wanted to give you an idea on what we do and how we measure it, and making sure that you understand that also that's also safe. This is not crazy guinea pig type research. So I also mentioned that we were able to measure how the muscle, the heart, and the skeletal muscle is able to produce energy, and we're able to do that through measurement through an MRI machine, which I'm sure many of you have at least had some experience with. And this crazy device here is our exercise device. So the participant comes in and actually pushes down on this device. It's like a gas pedal that has a pneumatic resistance chamber here. And actually, so the MRI can actually, instead of looking at pictures of the muscle, it can actually measure concentrations of different metabolites inside the muscle, in the heart and skeletal muscle. And we can quantify basically how much energy the muscle and the heart can produce through the mitochondria. So aerobic energy of the skeletal muscle and of the heart. I'm just going to skip right through these things here. Okay. So where are we to date with this large study? So basically, we're pretty much finished. We studied 14 participants with Barth syndrome and 15 unaffected controlled children. We studied 15 Barth, young adult Barths, and 12 control adult unaffected adults. 4 international families participated, and we have had no adverse events or no issues at all in studying nearly 29 participants with Barth syndrome. Okay, so where does this bring us as far as the heart transplant study? Okay, so why did we want to do the transplant study? So as you probably, again, know that about 50% of those with Barth syndrome ultimately require a heart transplant. And in patients without Barth syndrome, those who do have heart transplant really improves a lot of things besides obviously the heart. Heart transplant in patients without Barth syndrome improves muscle mass. It can improve skeletal muscle mitochondrial function. It can improve vascular function, so blood vessel function, It also improves exercise tolerance and also can change skeletal muscle metabolism. So a lot of positive things in those who have had heart transplants can occur in those without Barth syndrome. But we really don't know what is the effect of having a heart transplant in Barth syndrome. How does it affect all these different things? How does it affect exercise tolerance? How does it affect metabolism, which we talked about? How does it affect energy production in the skeletal muscle? How does it affect exercise tolerance? How does it affect body composition? So all these things that we don't really understand in Barth syndrome, the effect of the heart transplant, we want to know about. So we think it's likely that after a heart transplant, many of these adaptations that we see in those who don't have Barth syndrome who had heart transplants could potentially be beneficial in Barth syndrome and affect this metabolic phenotype that we're talking about. Let's say metabolic phenotype, talking about how the heart and skeletal muscle use nutrients, how they produce energy, exercise tolerance, muscle strength, muscle mass, all those types of things. So what we want to do is characterize this metabolic phenotype. And we thought that we can characterize this in those who've had transplants and compare them to those who didn't have transplants, the study that we just talked about. We could really provide us with important information that might lead to unique treatment paradigms or tailored interventions or some sort of potential dietary intervention that might benefit those with transplants. So this heart study transplant aims are to first characterize peak exercise tolerance, and that basically, which we talked about, is to do a peak exercise test. So how that goes is we explained a little bit before, but you basically get on an exercise bike, and every minute it gets harder and harder to pedal, and we measure your peak oxygen consumption. So basically, you're going to be breathing in and out of a mouthpiece when having an EKG on there on your chest, and basically we poop you out. We increase the resistance of the bike until you cannot pedal anymore. The test takes anywhere between 8 and 12 minutes. And so I think you saw a couple pictures that we were able to do that this for, and some of you might have undergone this already. Our second aim is we want to characterize skeletal muscle morphology and strength. So this is a little bit different that we did in the main study, but we wanted to look at muscle mass, so through body composition, which we did do in the initial study, but we also want to measure strength, which we've been doing through our resistance training protocol. We're interested in not only muscle mass changes, but also muscle strength changes that comes with having a heart transplant. So the two procedures that we're going to be doing there are body composition, which basically all it is going inside of this device called a bod pod, and it looks like most of you are probably too young to remember the show Mork and Mindy, but he used to be transported into this pod-like device. And it takes about 7 to 10 minutes, and all you do is sit in this chamber that looks like a big egg, and just basically remain still. Then we do some strength testing, which is going to basically measure your muscle strength through 1 rep maximal testing at certain devices that we have at WashU. And then the third aim is to characterize heart and skeletal muscle substrate metabolism and energetics. And these are the procedures that I just talked about. This clinical metabolism study where we give fat and sugar tracers during that 3.5-hour study, during 2 hours of rest, half an hour exercise, and then 1 hour recovery, where we take a lot of blood and breath sample collections, so we can tell a lot about metabolism. And you're also going to have the magnetic resonance imaging study, so the MRI and echocardiography. So again, we're going to compare all these things that we measure here and compare them to the initial study, which I talked about in participants with Barth syndrome who did not have a heart transplant. So what we're doing is basically adding a transplanted group to our existing study, which I talked about. We're going to compare the two groups. So they're basically those who participate in the transplant study are going to participate in the exact same protocol and procedures as those who didn't have a heart transplant, except we're not doing the PET imaging study, which I really didn't talk about anyway with that. So you basically be doing everything that the original study has done. Also, we'll participate in an extra strength testing procedure. Again, doesn't take very long, but it's just something we wanted to add on to look at strength testing. The study takes about 2.5 days. So again, we will fly you to St. Louis. We'll pay for the flights and the lodging and local transportation for both the participant and the parent. Again, if the parent is a child or if someone wants to bring someone anyway, we'll pay for both all this for both parent and the participant. We'll also give a $300 remuneration for your time and your effort for the study as well. So who are we looking for? So we're looking for boys and young men with Barth syndrome who had a heart transplant. So as young as 8 and as old as 36 years. So really looking at through the whole age spectrum. And the reason we're looking at that is that was basically the age requirements of the original study. So we've studied guys as young as 8 years old in this study. So they're able to participate well with these procedures with really no problem. So the only thing we're asking for is having a heart transplant greater than 1 year. So there's no lengthwise. If you had one for our original criteria was that those who had a heart transplant greater than 10 years would be excluded, but we're not doing that. So you can have heart transplant 15 or more years. So that's not an exclusion criteria anymore. So we ask you probably just for a logistics standpoint, living in North America, UK, or Europe. Or if there's some ability to travel to the U.S., okay, we can. So basically, we would take almost anyone, wherever they live. We ask that you're stable on medications for greater or equal to 3 months. Being sedentary, so exercising less than 2 times a week. And then the only exclusion criteria that we have is if you have known active acute rejection. We'll probably just wait until that rejection has subsided to study you. Or any known moderate or high vasculopathy, coronary or allograft vasculopathy of the transplant. If you have a presence of cardiac pacemaker or ICD, we could probably still study you, but please contact me about that if you do, because that's an exclusion criteria, obviously, for the MRI. So what are the potential benefits of this study? Well, we hope it'll help us further understand really the pathophysiology, the cause of disease in Barth syndrome. We also think that it potentially may help identify unique treatment needs of those with heart transplant. That's really the ultimate goal of this. And if there's something that we're able to help the guys with transplant or really treat them a little bit differently or more effectively, that we're hoping that some of this information will help us do that. Potentially, this may point to an optimal diet and exercise program, specifically for those who had heart transplants. We also hope this will validate outcomes for potential clinical trials and may point to unique outcome variables for transplanted patients, because obviously we want to include guys that had transplants in some of these different clinical trials that we're developing. And then again, it provides evidence to the NIH that the families with Barth syndrome will participate in these clinical studies, flying to St. Louis, giving your time for 3 days, undergoing these more invasive type studies. And so far, we've done that. The Barth syndrome community has been fantastic. So this is our research study team. Besides myself, Dominic Reeds, who probably many of you have met at the last year's conference, and Kay as well. So Dominic is an endocrinologist here at Washington University. He basically provides general medical oversight here. Kay is our wonderful research coordinator. So again, many of you probably have met. Linda Peterson is a cardiologist, kind of oversees our studies. Adil Bashir, he basically helps us with the MRI, energetic data. Everyone knows Barry Byrne at the University of Florida and Christy Pasak there as well. So we have a really neat team involved in this project. So this is a picture of WashU. There's a lot of people that are also involved in this, and a lot of different funding that'll help us do this study. This is Washington University in St. Louis. And we just really want to thank the research participants that come and bring their family and spend a few days in St. Louis with us and really donate their time and their effort to really learn more about Barth syndrome, so we can really ultimately develop effective interventions or hopefully potentially a cure for Barth syndrome. So with that, I'm sure there'll be many questions, and I'll be happy to take questions. Okay, so I see one question.
There's several questions. I just sent you there are 1, 2, 3, 4 for you to start with, and I'll keep copying. So if you want to start from the
So
bottom and then I'll continue.
is it 738? Is that the, oh, that's the last one. Let me see. Why don't I,
Uh,
I'll just scroll down here.
yeah.
Uh,
So I'll just go for it. It says, will it be possible to compare the data collected during previous conferences prior to transplant with this study?
Absolutely. So if previous conferences or studies that were, you know, if the procedures were the same, absolutely. So like exercise testing, absolutely. If we were able to, if we did like some sort of metabolic study or body composition or strength, absolutely, we can certainly compare the pre and post transplant data. In fact, that would be very, very interesting if someone didn't have a transplant and was tested, and then had a transplant and was tested after. That would be obviously very, very informative. Okay, I think I
Okay,
can.
next one. So the next one is, my son won't be 8 until February of next year. Can he participate in this study?
Absolutely. So we just have to wait until next February. So this study will probably be going on for, my guess is 2 years, just to get everyone in. So absolutely.
You were so fast. I can't keep up with you. As I
I'm,
was saying, I was telling everybody, come
that
up
was
for
an easy.
air. At some point, he's going to come up for air. Okay, so the next question is, has there been any results from the original study that can be discussed, and how did those support the initial hypothesis?
Oh, that's a great question.
Oh,
So
you've
the
got
answer is
some more
yes.
coming up.
Great question. So the answer is yes. We published one paper that is open access just several months ago, looking at the heart and skeletal muscle energetic data that we're able to generate from the MRI. And those with Barth syndrome have significantly impaired ability to generate energy, basically through the mitochondrial energy production in the heart and skeletal muscle compared to their unaffected peers, both in adults and in children. So right now we're analyzing the metabolic data for publication, but what the metabolic data showed is that fatty acid metabolism is impaired, especially during low-level exercise in Barth syndrome. And what it seems to happen is that guys with Barth syndrome seem to compensate for this impairment in fat metabolism by increasing glucose metabolism. So it seems like because they're not able to use fat as well as their unaffected peers, that they compensate by using more glucose or sugar metabolism. So hopefully we're going to be submitting that for publication in the next month or two.
Okay, the next question is there a hypothesis why there is an improvement after heart transplantation?
That's a great question. So we don't know anything in Barth syndrome, but based on heart transplantation without Barth syndrome, it seems that the heart is able to, when you have a heart transplant, the heart function improves. So you're able to deliver more blood to skeletal muscles, and that could potentially improve muscle metabolism and muscle energy production. And that's mostly both based on animal work and some hypotheses, but that is the thought process why muscle metabolism improves when with people who have a heart transplant.
Okay, the other question was around slide 30, and the question was, are Barth guys without transplant, are Barth guys without transplant considered the control group? I guess for,
Yes,
I guess, and that's how it's written. So
yeah.
if there's clarification, we can get.
That's a great question. So yes, there actually will be two control groups. So we're able to, because the initial study compared guys with Barth syndrome without a heart transplant to unaffected controls, we're able to now compare guys with a heart transplant to guys without a heart transplant and unaffected controls. So we're actually able to look at three groups. So for instance, do guys with heart transplants, did they look more like guys with Barth syndrome without heart transplants, or did they actually look more like unaffected controls? Does it basically normalize their metabolism and things like that? So the answer is yes, both. There'll be two control groups actually, unaffected peers, and then the Barth syndrome without heart transplants. Is that clear? Does that make sense?
It did to me. If the person who asked the question has any other questions, you could send it now. Is there? Yep, it was clear. He said, "Are there any other questions? If somebody has a question and you're in the midst of typing it, if you can just raise your hand so we know to wait for you." And then we'll just, and of course my phone's ringing. It's better dogs barking, but if anybody has any questions, if you can just type them in or raise your hands. If you can just wait a second, Todd, and let's just make sure that nobody has any questions.
Sure. I'm not going anywhere. So one thing, if someone's really interested in getting the real life scoop on what it's like to participate in this study, I could ask some of our participants to reach out to whoever's interested to kind of get a behind the scenes, what is it really like when I'm not listening type of response.
Todd, this is Matt. Can you hear me?
Yeah, I can hear you.
Okay, good. Excellent talk. The one question I have is, could you explain why the heart is different from the skeletal muscle, or what's the idea of why that they're somewhat different? You mentioned before that people with heart transplants get, they have certain improvements. Now, all the boys with heart transplants, they got a heart transplant because their heart was pretty bad off. So it wasn't, they didn't get transplanted because it would make them feel better. They were in pretty serious condition. So they got a heart transplant. But we know from other data that the heart and the skeletal muscle, even though these are both muscles, they're separable in certain ways. We've seen this with bezafibrate, where the heart function of the knockdown mouse gets better with bezafibrate treatment, but interestingly, their fatigue levels are the same. That does not improve. So I just thought that maybe you could talk a little bit about the heart, how it's different from skeletal muscle, how it's like skeletal muscle. And I just wanted to impress upon the audience that this is some fascinating, interesting scientific investigation that you're doing. And there is nobody like Barth syndrome boys that I know of. The metabolism that you've discovered and published upon, Todd, is unique. And having this third group, like you said, guys with transplanted hearts, is going to tell us not only a lot about Barth syndrome, but I think a lot about how the heart affects fatigue and how the heart is impacting other aspects of metabolism in the human body. So I think this is a superb and super critical study, obviously not only for our own community, but also for science in general, because the guys are just unique metabolically. And this will be, really interested in seeing what you find out here.
Yeah. So I'll try to answer some of the couple questions that you posed there, but thank you for that. And I think you're right. It's Barth syndrome is very unique, even in the whole realm of mitochondrial disease. It's very, very unique, and we're really just beginning to understand more about Barth syndrome. And I think this study will certainly help. But cardiac and skeletal muscle are similar in a sense where they're both striated muscle, which means they're able to basically generate high force muscle compared to like smooth muscle, like that line your GI tract and your blood vessels. They have numerous nuclei. They have they require a lot of energy, but heart's a little different than skeletal muscle. If you think about the function of it, hearts beat, anywhere from 60 to 100 times a minute, where skeletal muscle does not have that metabolic demand on it like the heart does. And the heart generates most of its energy from aerobic energy production, i.e. through the mitochondria. And so potentially why the heart potentially gets more affected in Barth syndrome, or that's why guys have to get heart transplants or you have these crashes, is that the heart requires such a constant flow of energy from the mitochondria that's why the heart seems to be affected potentially more so than the skeletal muscle, or at least functionally, because it requires so much energy, where the skeletal muscle only requires energy when you're walking around or going upstairs or whatever it may be. So it's going to be really interesting to see if a healthy heart or an improved heart can affect skeletal muscle metabolism or a whole body function. Can it improve fatigue levels? Can it improve exercise tolerance? Can it improve muscle metabolism? Can it improve body composition? All these things that we really don't understand. So I think that this study will really help us kind of unravel all those questions.
Yeah, great. We have a question here. Basically, it says, when can we start? So what's your timeline for starting? I know you have approvals and everything like that. When is your timeline to start things?
We are ready to go. So I'm going to actually go one more slide here, and I'm going to provide our contact information, and you can contact myself or Kay, and we can get you scheduled as soon as you're able to do it, and we can coordinate our schedules and everything, that we can get it started right away.
Oh, we have another question here. One of the individuals has a diaphragm pilication, Pilication, on one side. The oxygen sats are at 97%. I guess the idea is that will that affect the breathing test that you do, the breath analysis?
It will not affect the breath analysis. Obviously, that might affect exercise tolerance, but I don't think that that's an exclusion criteria.
Okay, right. Okay. Anybody else have any more questions? I know Shelly had to step away for a few minutes here.
I'd be more than happy if anyone's interested in emailing me or calling me, has more questions about the tracers or any of the procedures. I know I kind of quickly flew through them. I didn't want to get into too much detail to get confusing, although it might have been too superficial. So I'm more than willing to talk on the phone or through email to explain more about what these tests involve.
Yeah, we have a question also. Can Todd explain the difference between his two studies? Two studies. Basically, you're just doing the same study with guys that have heart transplants, and that's the real beauty of it, because you can directly compare to the guys who don't have heart transplants, as well as the control group without unaffected individuals. But
so, do they mean the two studies, or they mean the other resistance training study?
I'm not sure. It just says exercise versus metabolic phenotype.
Yeah. So the metabolic phenotype. So the exercise study is an exercise training study, and we have not expanded that to the heart transplant guys yet. Although that's probably something that we want to do down the road. But what we're doing now is basically looking at the effects of a strength training program on guys with Barth syndrome, and looking at the effects of things like exercise tolerance, muscle strength, body composition, things like that. But with that, hopefully we can expand this to guys with transplants. And I think this initial characterization study will really kind of guide where we go with the exercise training studies.
Todd, I have my own personal question. I'm always interested in using exercise to treat disease. And there are some diseases where physicians don't recommend any sort of exercise, which just seems counterintuitive to me. And what I've read in the literature, especially with Duchenne muscular dystrophy, is that you're not supposed to exercise because it's not good. And then I read reports, publications that it's actually good to exercise with Duchenne muscular dystrophy. Now, of course, you've been doing exercise testing and exercise training on many of the guys over the years, and it's at worst, not bad. And at best, it could be really very important in improving your health. Could you give us a little, do you have any thoughts about that in general? Because in the specific case of Barth syndrome, I don't think there's any question that exercise is not an impediment.
Yeah. So in Duchenne, the thinking, the clinical thinking on the initial thoughts that exercise was bad for you is changing. So initially in Duchenne muscular dystrophy, they thought that if you exercise too hard, that you induce fibrosis in the muscle, which is a big problem with it because it's irreversible in Duchenne. But now they're finding that actually if you keep the exercise to a moderate to lower level, that is actually beneficial in strength and function in Duchenne. So that's changing a little bit. But what we've found so far in Barth syndrome, first of all, both aerobic exercise, so riding bikes or treadmill or swimming or whatever, seems to be completely safe in Barth syndrome. Also resistance training, so strength training, has also been very safe in this population. So we're as far as safety goes, we're not worried about it at all. And it seems like so we initially did some aerobic exercise in guys with Barth syndrome. It seemed to help some, but not as much as we hoped. And we thought potentially the reason is because with aerobic exercise, when you do like things like riding the bike or these other elliptical and things, what happens is when you do this for long periods of time, you increase the number of mitochondria and the efficiency of the mitochondria. We thought in Barth syndrome potentially just making more mitochondria that are not working so well. So that's why we switched to strength training to try to really target types of muscle that seem to be less reliant on mitochondrial metabolism, to really strengthen and really increase the effectiveness of those types of muscles to improve exercise tolerance, quality of life, strength, function, things like that. And that's why we kind of are looking now towards the resistance. I think in the end, it's probably going to be a combination of the two that's going to be most effective, but we really don't know that yet. So we're continuing to do those studies.
Okay. I think Shelly's back, if she can come back in, and I don't see any other questions from the group unless
yeah, I am back. Thank you. I had a call I had to go away from, and I missed something. I think Matt, you said the question about the diaphragm was answered.
I think Todd answered that pretty well.
Okay. Todd, this is really exciting. I know that the transplant families and the guys have been more than willing to participate in research and have been kind of sitting on the sidelines. And this is going to be so interesting to see what it looks like with a transplant. And I think it will be very, very helpful too, because we are still encountering challenges with kids being listed for heart transplants because of the condition. But we see the guys doing really well with the transplants and thriving. So I appreciate you looking at this segment of our population to get the word out about this.
Well, I'm thrilled because I felt so horribly the last 5 years that I have to say that guys, with this study, guys with transplant are excluded. So I'm so happy to to do this. And also, I think it's going to be a great first step of just kind of what do these guys look like as far as exercise tolerance and all these things we're measuring compared to those who don't have transplants. A great first step on kind of leading toward treatments.
Yeah, it's sort of like turnabout. Before, all the transplanted guys could not volunteer for stuff. They were excluded. Now, the only people that can get in this study have to have a heart transplant. So turnabout is fair play, and it's going to be such an interesting study to see the results and then communicate that to the wider scientific world. I think it's fabulous.
Todd, I do want to clarify something. So initially we had some miscommunication, and I just want to clear this up just in case anybody sees this webinar and is thinking that there is a max time frame. I may have missed it, but I don't think that you mentioned that there was a cap on the amount of time that they had to be post-transplant. Initially, it was 10 years. Could you explain a little bit about why we were looking at a cap, or is there a cap on how long they can be post-transplant, or at any point can they participate in the study?
Thanks for clarifying, and I did try to clarify that during the presentation. So there is no cap. So they can be 10 years, 15 years, 20 years is totally fine. I think we initially just put that in there to make sure that they didn't have participants didn't have severe vasculopathy that tends to happen many times with long-term transplants. So Brian Feingold is collaborating, Carolyn Taylor is collaborating on this study as well, and they were the ones that initially said it, but we went back to them, and they are completely fine with eliminating the cap. So there's no cap. The only criteria is that we want them to be a 1 year post-transplant, 1 year or greater post-transplant.
Thanks for clarifying that. I didn't want an old message. You know how you read one message and then it just, that's the one that seems to stick in everybody's file. So unless anybody, and you're going to have to raise your hands really quickly if you're about to type something, but unless anybody raises their hands on their dashboard, I think we can close it out. It's 9 o'clock. So Todd has given you the information on his on the last slide of this folder where you can contact him. You can also go to barthsyndrome.org and click on the link for opportunities to participate in research, and we will have the information, Kay's contact information and Todd's contact information to participate in the study. As he said, he's ready to go. You can contact people. You can contact Todd, Cade tonight and let them know that you're ready to go and want to participate. I think there's already a couple people who are signed up for the study. And I just appreciate everybody stepping up and being so willing to do this. This may well be the fastest enrollment we have ever had in any study ever. So way to go, transplant folks. So I think everybody's saying thank you, thank you, thank you. Waited for this opportunity for a long time and we're so excited. Thank you, Todd. Thank you, Todd, for doing this. And we will see you later. I'm going to go ahead and get this ready and have it up on the web this evening. So thanks, Todd, so much. And thanks, Matt, for taking over for me. And thank you, everybody, for being here.
Well, thank you, everyone. And thank you, Shelley and Matt.